And so I got involved in the COVID sort of dissonance side of things because when I saw how they were using PCR in COVID, I realized it was a fraud. Like we run PCR on cannabis plants all day long. This is our bread and butter. We make tests that look for pathogens on cannabis. Like that
PCR is essentially like the nose swab test that people are doing.
Yeah, and PCR is actually a really good test. They destroyed the reputation of PCR in COVID. You know, it can
This was invented by Kerry Mullis?
Yes, yeah. Great book. You should read his book.
I've been wanting to. I've watched some interviews with him. He seems like a really cool dude. He was a surfer.
He was a surfer. Sadly, died right before the pandemic. But
and he hated Fauci.
Yes, I think anyone who gets their nose into the space will come to the same conclusion as him. So, it's unfortunately, if you're in the public, he's elevated as some almost Jesus level saint. But if you see what he actually does behind the scenes, he is the exact opposite of that. So the pandemic comes along, they're abusing these PCR tools, and I make note of this. I write a paper about it, and of course it gets attacked. It goes through the typical censorship and ridicule that you expect. And shortly after that I think that caught the attention of Peter McCullough. He asked me to help write him a paper about the differences between the vaccines and the virus, which I went through all the molecular biology and how those two things differ. And after that happened, people start randomly sending me vials of vaccines to our address, our company.
What prompted you to first write the paper?
Well, I could tell that since the PCR was a fraud, that when I saw what they were going to do with the vaccine, I'm like, this has not been tested. I know how that sausage is
vaccine.
made. Yeah, my history of building DNA sequencers meant I had a deep respect for when you start changing nucleotide chemistry on RNA molecules, you have no idea what you're doing. And that's what they did in the vaccine, is they changed every single uracil out with pseudouridine. That completely changes the function of that RNA. And these guys just said, oh, we're just going to change all of them and then inject it and see what happens. And you change that one methyl group in the course of trying to build DNA sequencers, and your whole sequencer will fall apart. I mean, the enzymes are that sensitive to a single methyl group being placed on some of these pieces of DNA. You put 800 of them on the RNA that you're doing, you are in completely unknown territory with how that RNA is going to fold, how it's going to be processed in the cell, the RNA interference pathways it's going to kick off. So when I saw what they're doing, I'm like, this is complete reckless insanity that they're doing, and no one's raising their hand about it, which is frustrating because you're in a field and you look around and none of your peers are talking about this, and you're like, is everyone in on this? Like, what's going on? And being in Massachusetts, pretty much everyone is in on it. Pfizer's there, Moderna's there.
Oh, really?
I mean, the PCR companies are all there. So it's a breadbasket of people benefiting from the pandemic.
So basically, of comparing how sloppy the PCR test would be to measure this, you assumed that they were going to be way off with the vaccine. Is that a good way to sum it up?
Yeah,
okay.
I could see that there was a game going on here to pump up the pandemic's risk so that there would be a need and a public desire for a vaccine. If you actually paid attention to how the PCR was behaving and how they were overcalling this disease, you would have barely have noticed this pandemic. Like the COVID pandemic that came through was predominantly human-induced death. It was not that people weren't dying from the virus. They're dying from what people's reaction to the virus. Like they're putting them on remdesivir, putting them on ventilators, taking away antibiotics, not letting them use ivermectin. There was all types of iatrogenic death, if you will, from how they handled the pandemic. But very few people actually died of the actual virus.
The people that died actually had lots of comorbidities, right?
That's
and the virus is basically like the final straw that broke the camel's back.
And it just happens to be there. And the hospitals were also all incentivized to tag everyone they could with COVID and give it to them on the way out the door so they could get better billing, right? So if when they were PCRing corpses, I mean, because if you could find COVID, you had no liability for the patient and you got a completely different reimbursement structure.
What?
Yes.
They were actually doing the tests on corpses.
Yes, they're corpses, and there's John Bodwin you should talk to. He FOIA'd all the death records in Massachusetts, and all the people who are labeling these deaths of people that were clearly killed from the vaccine, they label as COVID.
Wow.
So the whole COVID thing has a layer of scam to it. Now, that doesn't mean that the virus doesn't exist and that it doesn't get you sick. I got it. I got horribly sick. I had a really long recovery from it, but it didn't create that excess mortality wave. That was created by people's reaction to it. So I knew that when the vaccine came out. I'm like, okay, this vaccine, they've got the whole population primed to be scared out of their mind that they're all going to take this reckless drug that they've they've barely studied, and that's going to create another wave. And I think there's probably more people dead from that than there is from the virus. At least if you go through John's work, there's easily 550,000 people that are dead in the United States from the vaccine. I don't think the virus comes anywhere close to that. They'll tell you a couple million people, but 90% of them are comorbid, and then you start itemizing all the other types of iatrogenic death in there. It's probably a tenth of that. It's probably fivefold less than the vaccine death.
Okay.
So anyway, these people, after I wrote this paper with Peter McCullough, people started sending vaccine vials to my company address, which it's like,
who sent you? Who got a hold of these and sent them to you?
I don't know. They came anonymous.
Who could it have been?
Probably my Twitter followers.
How would they get their hands on vaccine? They just work at a pharmacy or something?
I'm sure if they got their hands on them, they don't want to tell me how.
Right. I'm just trying to figure out how they would have done that.
Oh yeah, pharmacists have them. They're around. They throw them out. So the ones that we got were never opened. So
oh wow.
So
okay.
they came. They have these tamper-resistant seals on them, and so you can tell whether they've been opened or not. And we got some that were expired and some that weren't expired. But when I kept getting them, I just kept throwing them in the freezer, being I don't know what to do with these things. But and then this cannabis experiment I mentioned earlier was like, okay, we have a problem. I need a pharmaceutical grade vaccine. Maybe I need pharmaceutical grade mRNA. And I'm like, these things are sitting in the freezer. Just spike one of those in. It's got a poly A tail that should tell us what's going on with the RNA-seq that we're doing, and it did. But out came the vector that they used to manufacture these things, and that shouldn't be in there.
The vector that they use to manufacture. What do you mean by that?
So when they make these mRNAs, they have to have a DNA template to make it from. So all RNA is actually synthesized usually from some other piece of DNA. So,
okay.
So they build a piece of DNA that is think of it as like a mold. If you're into injection molding, you need something to kind of print the RNA. The DNA they have to make it in the form of DNA first. So they take that spike protein sequence and they first code it into DNA, and then they use an enzyme known as T7 polymerase to copy the DNA and turn it into RNA. And when they're doing that process, in the case of these mRNA vaccines, they rip out one of the bases and put in this pseudouridine, so that there's only a pseudouridine and there's no uracil. That's how they get these mRNAs made. Now, that means they have to have a piece of DNA that codes for the spike sequence they want to make turn into RNA, but they need something to propagate that DNA. The DNA is not going to organize itself. So what they do is they put a couple other genes on that DNA that make it so E. coli will replicate it.
So a what?
That E. coli, a bacteria.
Okay.
So basically you can brew it. And this is, we did this in the Human Genome Project, 20 million different clones. It's a very common biotech thing. What you do is you put an antibiotic resistance gene attached to the spike sequence. And that antibiotic resistance gene and a couple other pieces of sequence guarantee that it will grow inside of E. coli. So when you put that DNA into E. coli, you can then brew up huge vats of E. coli, and now it's copying your plasmid as long as there is a piece, there's antibiotics in there that match the gene. So in this case, they were using, what do they have? I think they use kanamycin in this case. Kanamycin is an antibiotic. So they have a spike sequence attached to a kanamycin resistance gene, attached to what's known as a bacterial origin of replication that E. coli will grab onto and replicate this DNA. Once that's in a bacterial cell, you just have to grow that stuff at 37 degrees overnight, and you get millions of fold amplification of the cells. They double every 20 or 30 minutes. And so every time the cell doubles, you get about 200 copies of that piece of DNA with it. So fermenting this overnight, and suddenly you get yourself millions and millions of copies of this DNA that you now need to get out of E. coli. So you lice it open with some soap and purify that DNA, hopefully away from all the other junk. And now you've got your plasmid purified, ready to turn into RNA. That's kind of the manufacturing process that they use for making these Pfizer vaccines. Now,
what is a plasmid?
A plasmid's that circular piece of DNA that kind of carries the spike DNA that you want to have replicated in E. coli. So the plasmid is circular. So it contains an antibiotic resistance gene and an origin of replication that ensures the E. coli cell will replicate it. So if you glue your spike sequence onto that piece of DNA into a circle, put that in E. coli, and give it an antibiotic, the only E. coli that will survive are the E. coli cells that have the plasmid that code for the resistance.
Okay.
Oh, there you go. Nice. Plasmid maps. So, You can see a bacterial DNA in there. The bacterial DNA in E. coli is like 6 million letters long. These plasmids are like 5,000 to 10,000 letters long.
Okay.
But they're circular. That helps them replicate, and through a process known as rolling circle amplification, but also keeps them from degrading as quickly because there's no ends of DNA. When they're tied in a Mobius strip like that, the enzymes don't know how to really destroy them. So plasmids tend to stick around for a very long period of time. But on the backbone of that plasmid, you will have an antibiotic resistance gene. I think you had another one over there that was like a plasmid map. If you just scroll one image over, like there you go. Bang. If you click on that, you'll see they have these little pieces on them. So the inserted gene would be the spike that they put in there.
The inserted gene is the spike. And what is the spike?
The spike is the sequence that they want to make RNA for the vaccine. That when it gets into a mammalian cell, will create a spike protein that your immune system theoretically will learn how to fight off COVID.
Got it. Okay.
There's a huge leap between A and Z there. Okay, that we'll go into.
Okay. I'm trying my best to keep up with you here.
Yeah. So that inserted gene ends up being, they put in the spike sequence there. There's an origin of replication down there that teaches the E. coli cell to, hey, copy this circle over and over again if you ever see me. It basically recruits a polymerase to do that.
Okay.
That antibiotic resistance gene, let's say that's kanamycin in the case of Pfizer's vaccine. That means if the coli is growing in kanamycin, the only cells that can survive are the ones that have soaked up the plasmid. So it's a selectable marker, which means the only E. coli that brew are ones that contain your DNA. Otherwise, when the cells divide, your plasmid just gets lost. So that selectable marker is really critical to make sure the plasmid sticks around when you grow an E. coli. But once you have your gene in a system like this, all you need to do to get the E. coli is you heat E. coli to like 37 degrees and it gets porous and it soaks up the plasmids. And then it has the superpower of being resistant to kanamycin. And so now only the cells that soaked up the DNA can survive in the growth. They start replicating, and now they're replicating your hijacked spike sequence with this.
Okay.
So the challenge here is that you now don't just have your spike sequence as part of the contaminant in your vaccine. You have all that other crap, the antibiotic resistance gene and all the origins of replication that you introduced as replicative machinery to manufacture your...
But they know this, right? They know that this stuff's going to be in there.
They know it.
And do they have a process to get rid of it?
They try. They fail. But that's the point where they kind of...
Is this in all vaccines?
No,
only mRNA.
These mRNA ones are very unique, right? We've never done vaccines like this before.
Okay. How many mRNA vaccines do we have?
We have Moderna and we got Pfizer.
I mean, other than the COVID.
They're just rolling out RSV and I think flu ones now.
Okay.
There's really scary ones going on in Japan right now. They're trialing out a self-amplifying mRNA vaccine, which is horrifying. That is something that's likely going to spread out of control. So there's not many.
So COVID was the first one we had.
Yes.
Okay.
First one we had.
Okay. Okay, but so obviously they knew that this stuff was going to be in there because that was purposeful. You needed that to actually create it and for it to replicate.
Yes.
And they didn't have a good enough protocol to get rid of it.
Moderna had a better protocol. In fact, what's really key about this whole story is if you read Moderna's patents, they speak to this problem. That one, it's very difficult to figure out how much of this is left behind with PCR alone. So Stefan Bancel has a patent that says don't use qPCR to measure what's left behind. But they also have some other patents in there on techniques to get rid of it. And the reason it is so important to get rid of is they write in that patent showing that it's oncogenic. If you leave this DNA around, it will insert into your genome and can create cancer. So you have to get rid of it. And that's written straight into their own patent estate.
So is this the same story that Jack Kruse was telling us about SV40?
It is the same story because the origin of replication that they have in Pfizer is an SV40 origin of replication. And they also have, you'll see that little promoter thing up there in pink. They have another promoter over by your antibiotic resistance gene and your selectable marker. They have multiple promoters on the Pfizer one. There's one over by that green arrow that is an SV40 promoter, and those elements are from contaminated the polio vaccines. Now,
right.
The polio vaccines were contaminated with the full virus. It was like 5,500 letter viruses. But if you study that virus, the most carcinogenic aspects of it are the things that are in Pfizer. They are basically the SV40 enhancer and promoter and the origin of replication. So the parts that are the most carcinogenic are in there, and they hid them from the regulators. When they were asked to, when they submitted this to the FDA demands that you give them a map like this that tells them what everything does. Like, show us where every selectable marker is, where every promoter is, where every open reading frame is. You have to disclose it all. And the one area that Pfizer refused to disclose was the SV40 components.
Okay. So essentially when Jack was in here, he was telling the story about how this guy named Alt Noshner was the one who created the polio vaccine. He famously went in front of a board of people or in an auditorium full of people and injected his grandchildren with it.
Nuts.
And then one of them died and one of them like lost a limb or something crazy.
Yeah.
And they found out that this SV40 came from, they were growing the vaccine on monkey kidneys or something like that.
Yep. Simian virus.
Simian virus number 40. And there was a lot of simian viruses, right?
At least 40 of them.
At least 40 of them. And then number 40, for some reason, that one created these quote unquote turbo cancers in humans.
Yes. Now that was the story about the LINAC. They were using the LINAC to actually do mutagenesis on the viruses to mutate them to see if they could get ones that were more potent as a bioweapon.
Jesus Christ.
And so, and they were planning to kill Castro or something with it, right?
Right.
It was a crazy story. I didn't know about that when I stumbled into this. I sadly learned about that after the fact and questioned whether I'd actually would have ran away and hid in the mountains if I had known that prior to disclosing this. But it is important to know we don't have the whole virus here. We have components in the virus here that I think are actually still very problematic and dangerous, but we don't have as firm of a grip on, is this causing the turbo cancers that people are claiming to come out of this vaccine? I think that's a more complicated story, that there's a Swiss Army knife of problems in the vaccines that can drive cancer, and the SV40 may just be one component of it, one hair that breaks the camel's back, if you will. There's issues with the LNPs. There's issues with that base I mentioned, that pseudouridine base, that completely scrambles your immune system. And then there's issues with repeat injecting this on people, that your immune system starts to become tolerant of pathogens when you do that. There's this thing known as an IgG4 class switch that happens with these vaccines. So there's a fourth thing is the actual spike protein itself has been shown to inhibit P53, which is a tumor suppressor gene that we have that cleans up your genome. So the protein that this plasmid's encoding shuts down your tumor surveillance. On top of that, the SV40 we know binds to P53, so that can't be good. You're shutting down the expression of P53 while you're also bombarding it with this SV40 DNA that probably makes it less functional. So it's a multiple hit problem. We've got multiple things that are wrong with these vaccines that are probably driving cancer. But what's annoying about the SV40 stuff, it's completely preventable, and they hit it, and it didn't need to be there. So that's just a sign that the regulatory structure we have here is completely captured. They are not. This didn't take. A cannabis company shouldn't be finding this, right? Like, this is something the FDA should have sequenced the vaccines. It doesn't cost much to do this anymore. When we started human genome, way back when they came up with guidelines for vaccines to have no more than 10 picograms of DNA in vaccines that you inject in your arm. Those are traditional vaccines. When you
10 picograms of DNA.
Yes.
Okay.
Very small amount, but your body, when you inject DNA naked like that, it's like a 10-minute half-life. It chews the stuff up really quickly.
Okay.
But we're now in a new world where we have lipid nanoparticles protecting that material, so it doesn't get chewed up. And those, so arguably the number should be like attograms. And the other thing that's happened in that time frame is they have dilated the amount of nanograms the pharmaceutical industry can have in vaccines 1,000-fold. So now it's up to 10 nanograms, because since the pharmaceutical companies back in 1986 got a liability waiver, right? Mm-hmm. So now they're not liable. Every year they lobby for looser and looser regulations, and they always get them. And so they've gotten to the point where they can put in 10 nanograms of DNA. On top of that, now it's on lipid nanoparticles, so it doesn't just get eaten up; it goes right to your cells. And they're playing around with putting DNA in there that's replicatable once it gets inside of your cells. So you could sneak one molecule through and it's a problem, right? So they really have opened up the tolerance levels here to extremely irresponsible levels, and they're not sequencing everything when the cost of sequencing has come down 100,000-fold in that same time frame. Like, they should know every single molecule that's there. There's no reason they can't know that with the tools that we've built. So as someone who's built sequencers, I get really pissed. I look at this and I'm like, this is utterly irresponsible. You could have sequenced every single vial that went out the door for pennies. You didn't do it.
And what is the thing? The reason that you say they purposely hid the SV40 in these vaccine vials from the FDA?
Yes, FDA has actually admitted to that now. And two other Health Canada's admitted it. TGA's admitted it. The EMA's admitted it.
And what do you think the reason for hiding that is? Do you think it's nefarious, or do you think it's just
intentional? It's intentional deception. Yeah, if they had that in there, it would not have gotten to market, and they would have maybe lost $100 billion.
And this was do you think this was about money, just Absolutely. To save money?
Yeah, if they showed them SV40, they would have been delayed at probably at least 6 months. They would have missed the market. Because that would have triggered alarm bells at the FDA that, oh my God, there's SV40 components in here. What are you doing? Oh, you got this from your gene therapy department? That's what they did, is in order to speed this up. Pfizer has a hilarious paper of them kind of chest pounding how wonderfully fast they released these vaccines, how they kind of built the plane while it was flying thing, right? One of these, self-backslapping type of papers. But if you go through that paper, you'll see that, hey, we were really quick at this because we have a gene therapy department. We were able to call them up and get these plasmids. That's where SV40 comes from. They use SV40 because it's a nuclear targeting sequence. It drives DNA right to the nucleus so that it can integrate. So this is a tool that's used for gene therapy, and Pfizer's like on record bragging about, yeah, we just lifted that and threw it into these COVID vaccines, and we're going to inject it into billions of people and see what happens, If the regulators saw SV40, I think they would have stopped and said, wait a minute, that's a nuclear targeting sequence. That's actually can replicate mammalian cells. You can't have something that replicates mammalian cells in your background, because that means 10 nanograms can become 100 nanograms or a microgram after it gets into a human cell. You have the machinery in there to teach human cells to keep replicating this. So once this came out, of course, they all claimed we were conspiracy theorists until the FDA went through their files and looked at the sequence Pfizer gave them and said, oh, wait a minute, it's in there. They just didn't point it out like they were supposed to. So their guidelines demand you submit the sequence. But
was this published anywhere?
Our stuff has been. Yeah, we've published it as a preprint. There's others that have since published the contamination story and gotten it through peer review. We're still forcing ours through peer review. It's going to be a fun journey, but others have gotten it through, and it's also been replicated. The thing about
I mean, the fact that what you were just saying, how the FDA found that there was SV40 in there, is that available online?
That is. I have the links to them on my Substack. Your group can look at all this communication. And a shout out to Scoops Magoo and Noah Chartier. They have been journalists who have actively been harassing the regulatory agencies with FOIAs and ATIPs to get this information out.
So what you were saying from your study on the cancer cells, sequencing the cancer cells, you were finding these things, these plasmids in the cancer cells. How much later? This was a year later.
Yeah, this person was 4x vaccinated and
4 times. Wow.
A year later. So.
And then what does that mean? What does that mean for them? The fact that it's still in these cells? Were these cells there prior, or were these cells created? And then what does this mean for people that aren't vaccinated, that are in proximity to these people?
All right, that's a couple points there. So so this patient's deceased, unfortunately. They have a year after vaccination, they got their first biopsy. A week after that, they got another biopsy, and then in 4 weeks they were dead.
Wow!
And they got a postmortem biopsy as well. We sequenced the postmortem in the first one, and we're looking at the differences between those two. There is a plasmid in there that codes for spike. It's the sequence of that spike is a little different. It's a lot different than the spike that Pfizer has in the vaccines we've sequenced previously. So this has raised a question as to what is really the source of this plasmid, because the differences there have raised alarm bells. Like, it's not exactly what we saw now we've only sequenced like 2 other lots before, so we don't have good surveillance of all the different lots for Pfizer. They could have different plasmids in each one. They could all be the same. We don't know. We need to do more to sort that out. But there is another hypothesis that could be at play here that leads to a much more frightening scenario, I think, in my mind, which is in the middle of the pandemic, there's a paper that was published that really went unnoticed. It was a group in Seattle that showed that their lab people were testing positive for COVID, but didn't have COVID. And they only tested positive. If you remember the COVID test, they actually had 3 assays usually testing for the virus. They would look at the spike, they'd look at nucleocapsid, and they'd look at the envelope region. So, and if all 3 lit up, they were pretty confident the virus was there. Occasionally, one of them would drop out, like the S target would drop out once in a while. They had that happen in Europe. It was called S target failure. And they would say, all right, we don't really know if you've got COVID because only 2 out of the 3 assays are working. So what was happening in Seattle in this lab is only the nucleocapsid test was going off, and that was really weird. And it was in all their lab staff. And it turned out that same lab was working on a vaccine that had nucleocapsid cloned into something very similar to Pfizer's plasmid there. So that somehow infected all the lab people, and then it got out of the lab and infected their housemates. And so the paper hypothesized that, okay, this must be because this is a shuttle vector that can grow in E. coli and can grow in mammalian cells. That means it's transmissible. our vaccine in the lab infected our staff and got out into the wild. This was another lab leak that didn't hit the radar anywhere. And they kind of wrote it off as, no worries, it's nucleocapsid, it's not spike. If it were spike, maybe we could have gotten those people some spikopathy, because the spike protein itself is really pro-inflammatory. Nucleocapsid, perhaps not as much. They kind of wrote the paper as, hey, watch out, your PCR test might be giving you false positives if you're working on these vaccines on the side. But for someone like me who reads that paper, I'm like, no, you just had a huge bio leak in Seattle, and it should have been followed up by the CDC. They should have run all over Seattle and looked for this, and all their housemates and everyone else. All right. So there are hundreds of labs around the world doing those same types of experiments where they're taking spike proteins that have been sequenced and there's some differences, and they want to characterize those differences, those variants of concern, right? You may have heard of this, like there's Omicron, Delta, Alpha.
Right.
All of these differences, there is a lab somewhere out there cloning that difference, putting it into one of these vectors and putting them into mammalian cells to see, okay, is it worse or is it better? And that's how they could gauge whether Omicron was like less pathogenic than let's say Alpha or Delta.
Okay.
So think of hundreds of labs around the world cloning, different forms of the spike protein into vectors like this that have the capacity to shuttle between mammals and E. coli. That means they're like little zoonotic bombs, if you will, that if they get into the lab and they get out, they could spread at their own unknown rate. We don't know what the R naught is of that type of transmission. It's not been followed. It's not been tracked very closely. But there is one case of it in the peer-reviewed literature already that it happened in Seattle. So that tells me if there's hundreds of other labs working on this, there could be leaks out there that we don't know about. So when I find something like this in a tumor biopsy that isn't identical to Pfizer, my head goes in that direction. Is that, okay, this plasmid that we found it in is called pcDNA3. It is the most commonly used vector for people to study spike protein mutations. So some lab could have leaked this and gotten to this patient somehow. You can't pin it 100% of Pfizer because it's not the fingerprint Pfizer gave to the FDA, unless Pfizer's playing with multiple plasmids, which their paper shows they are. They just haven't disclosed what they are. So we're stuck with two hypotheses here, that it could be something that is contamination in Pfizer's lab that got into this guy's 4 shots. He gets it. He ends up with tumor cancer a year later. Or it could be this person knew somebody at a lab and was in close proximity and got it from them. And that this ended up replicating in his body and ends up with in his colon cancer in some way. So we don't know, but I think the important message I want to get across to people is that the current gain-of-function debate isn't talking about this. The gain-of-function debate is people like Barrick making these full viruses. But all the research labs that just want to take a tiny piece of the virus out and say, I want to study what this Omicron thing is doing in the laboratory, they've got to be very careful if they put that into a shuttle vector that can infect humans and E. coli. That can't get out because the Seattle paper showed it can travel to housemates of the people working in the lab. So they have to make sure that when they do those studies that they have a kill switch in those plasmids to some extent that doesn't allow it to jump between different organisms. Like only put in a mammalian, something that can replicate in mammalian cells, or only E. coli but not both. Or they've got to just track them a lot more carefully. So, if Rand Paul's listening or anyone who's working on sort of the biosafety now thing, they have to look into that Seattle paper, and they have to take a look at the data that we found in this colon cancer thing to see, is it anywhere else? Like start using PCR to screen tumor biopsies. These tumor biopsies, people throw them out after 2 years. Unless they paraffin fix them, they can store them longer, but most biopsies are gone in 2 years. Pathologists don't keep them. So they've got to go and start looking to see, is this an N of 1, or are there other turbo cancers out there that are spike positive and have plasmids in them that are transmissible? Because if spike is in fact causing turbo cancers, they have a transmissible form of spike going through all the research laboratories right now.
So that essentially you're saying this SV40 turbo cancer would become contagious.
Well, it would move. Whether it would manifest in cancer in every patient is something I can't predict. Right. But they at least should be able to track it with PCR. You can go and track okay, is it leaking anywhere? Is it jumping from person to person? And are we finding it in more than just N of 1 biopsies here? We found it in both biopsy, but it was the same person, 2 different time points. But actually, we had 3 time points. They all had it. But the sequencing was only done on the first and last. But we do have to see if there's a trend here. If they keep finding this plasmid in other biopsies from people with turbo cancer, Okay, that starts to build a thesis here, and then they have to go and review which labs are making these. Did this come from Pfizer, or did this come from one of these labs that are studying VOCs? And what does that mean for how we think about gain of function research in the future? This paper has been tortured for years, and it's finally getting its way out in a journal without much change from the original work. And imagine if the world knew in 2021 about myocarditis, and this wasn't suppressed. All right, this signal now is. I was just with Peter 2 days ago on Saturday, and he gave a presentation on myocarditis and the fact that something they think they may have a better handle on this now, but they're finding ways to do a better job clearing the spike protein with a variety of things like bromelain and curcumin and I think the other one was nattokinase. But generally, in myocarditis, prognosis is like 50% fatality in 5 years, right? So, and they may have done this to hundreds of thousands of people. It's insane. And what you'll find the cover up that occurs is they'll say, well, the virus does that too. And the way they'll fabricate those stories is they'll do a publication showing that here's a population of people that got the virus and didn't get the virus, and they won't talk about the vaccine status in the paper. And they'll say, look, the people with the virus got more. Yeah, of course, if they got the virus and they got vaccinated, it's going to be worse. They got spiked twice.
Right. And what is the connection? What is the correlation with people who did get myocarditis? Was there any sort of pattern there?
Well, there was with the vaccine. It's now on the label. Like for the people who claim this isn't happening, like even the regulators have finally had to cough this one up. Like it's on the label.
It's on the COVID vaccine label.
Yeah, this can cause myocarditis. Like they've admitted to that on at the FDA site as well, right? That's ResearchGate, but the
Find that label, Steve-O.
The myocarditis story, I think why John's an interesting discussion on this is that he has the death records and he will tell you the myocarditis is the tip of the iceberg. There's way worse shit going on with this vaccine than myocarditis.
50% death rate is pretty substantial.
Yes, and Peter's been backing off of that now, being like they think they may have gotten a way to clear the spike protein now, that they can extend that. But before them having those treatment options, he was worried that was the prognosis of someone with myocarditis prior to them finding a way of eliminating some of the spike protein, but they are hopeful that number is going to go down with the treatments that they've come across. I think you'll probably find a notice from the FDA about myocarditis. I don't know if they'll actually fit it on those tiny labels.
Go to the FDA's website.
Yeah.
Yeah, but so
there you go. Update myocarditis.
Update on myocarditis. FDA. Okay, click on that. This came out in June 2022. Wow.
Yeah, it took them a while.
2 years ago. What is the summary of this? Go down.
This is the VRBAC committee.
That's a PDF.
Okay. Blah, 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 blah. What is is there like a summary or like a?
It's just like a brochure.
Keep going. Yeah, but keep scrolling. You'll find.
I've spoken at one of these VRBAC meetings. I think this is probably.
Okay, go up to that last one. Zoom in on the one with the chart on it, the red bars on it. Verified U.S. reports of VAERS of myocarditis after mRNA COVID-19 vaccination among people ages 18 years and older following primary series of first booster by time to symptom onset and dose number. Okay, so it's saying 1, 2, 3. That is the time to symptom onset.
Yeah.
And then the second column is verified reports. So the most verified reports is 3 days, and there's 250 reports. This is confusing. I don't know what this means.
Yeah, what you can see there is a dose response. I mean, that's what you'll see in Jessica's paper, and what Jessica and Peter did is they put this through what's known as the Bradford Hill criteria. So if you want to try and derive causation from correlative information, because that's where everyone goes to defend against this is it's just correlation,
right?
But it's a lot of correlation. So what Bradford Hill makes you go and do is you got to show that there's a temporal connection, that it's within close temporal proximity of the actual dose that this happens. You have to show a biological trend in that the more dose creates more problem. which they have here, right?
Okay.
So there's about 10 of these criteria in Bradford Hill that go through that help you determine whether something can be deemed causative or correlative. And they've satisfied Bradford Hill criteria on this one, on myocarditis. That's what her paper goes through. It's that, okay, there's a time connection, there's a biological mechanism, there's a dose response to this, and that got suppressed for 3 years.
Go to the next slide, Steve. 1,321 reports, what? Verified using CDC case definition.
Well, that's a word salad right there.
Yeah.
When CDC gets involved with case definition, they end up bearing most of it. But there are papers now out that look at autopsies that McCullough's been involved in, and it's something like that's the cold hard truth. And not enough autopsies were done. But when he was looking through autopsies, it was something like 70 to 75% of the cases they could pin to the vaccine. Whereas when it goes to the CDC's hands, it's like 7% or something. They start playing all these games, being like, well, it could have been something else. But kids don't get myocarditis. This is one that's really overt that they kind of had to cough up. I think where the cancer stuff gets messy is it tends to be sometimes older people, although that is not holding true with. I was just with Dr. Makis, and he was showing that like the peak of cancers were actually like in the 40s and 50s. So people are getting cancer like 10 years earlier. They're starting to screen earlier. Because of what's happened. Like they're trying to move the mammograms up 10 years. They're trying to move the colonoscopies up 10 years because that's, in some ways, that will actually hide the signal because you'll get more negatives when you do that. If you open up the scanning window, suddenly change it to 30 instead of 40 for breast cancer, suddenly you'll get a lot more people coming through that don't have it. And so the prevalence rate will go down if they just move the window around on you. But you are picking some people up at those ages, which you shouldn't be. That's what I think is the most shocking thing. And John's data shows us as well that the age demographic shift happened after the vaccine came into play. You could see when COVID was in place, the average age of death in Massachusetts was 82. All right, and the average age of death in Massachusetts is 80. So the vaccine was just the virus was just killing people on the edge of life. Okay. And Massachusetts data is really clear. And then when the vaccine actually comes into play, the people that are dying are suddenly 10 or 15 years younger. That's not, a virus doesn't do that. A virus doesn't suddenly mutate and decide to kill younger people.
Right.
That's something else that you've added in as a toxin that's bringing it. Not only does the age demographic shift in this data, the types of mechanisms of disease shifts from, in these older age patients that are getting hit with viruses, they're mostly going down with like some sort of secondary pneumonia. What's happening with the people that post-vaccine is you're getting clots, you're getting strokes, you're getting kidney failure, you're getting lymphomas. So it's the age demographic shifted and the type of disease shifted right after the vaccine program, which is a pretty strong signal that this is in fact not a virus changing on us in that regard.
And Jack said that aluminum ties into this story somehow. Is that true?
Well, there is some. So a person we work with on this was Sin Lee. Sin Lee is probably best known in this field for finding DNA contamination in the HPV vaccines. And I don't know that there's aluminum. I heard Jack say something about that. I have not measured for any aluminum in these vaccines, so I can't comment on that and whether it's in there. But the older vaccines do tend to have aluminum, and the problem with aluminum is it does bind DNA and pull it out of the solution. So sometimes the tools you use to measure DNA miss it. So it's a great way of camouflaging residual DNA in a vaccine is throw some aluminum in there, and all the aluminum soaks up the DNA so that the PCR tools don't see it. So Sin figured that out and was able to sequence parts of the HPV vaccines genetic constructs that they used to make it. And he's been complaining about that for probably over 5 or 6 years now. And again, the FDA just said it's move along, nothing to see here. But I don't know if that's playing any role in this event other than there is some literature out there regarding the efficiency of transfection. Lipid nanoparticles are really good at getting DNA into cells. In vaccines that don't have them, sometimes people use calcium phosphatase or other metals that will help move DNA into the cell. So there's some research that needs to be done as whether or not this aluminum DNA adjuvant actually has any level of transfection capacity. I suspect it's a lot lower than LNPs, but it may in fact facilitate some level of DNA entry into other cells. Ralph Barick's the guy who arguably taught Wiv how to create the coronavirus.
Taught who?
The Wuhan Institute of Virology.
Oh.
So there are grants that Fauci basically boomeranged off of EcoHealth into the WIV because they put a gain of function ban on funding here in the States.
Right.
And
Obama did that, right?
Obama did that. And so what Fauci's creative way around that was, well, we'll fund a nonprofit called EcoHealth Alliance run by Peter Daszak, who will launder the money for us into the Wuhan Institute of Virology. Right. Therefore, it's not directly from Fauci to Wuhan. It's going through some intermediary. And through that process, they taught the Wuhan Institute of Virology how to do this seamless no-seam technique, which is a technique of assembling viral genomes so you don't leave any evidence behind. It's a ligation technique that uses a Golden Gate assembly. That it's a clever way of putting stuff together so that you can't tell that someone built it necessarily from a laboratory. However, they did leave behind some fingerprints that make us that got them caught basically. And you should, if you want to hear about that story, you should get Charles Rixey on here. Charles Rixey has helped RFK write his book about this. And Charles Rixey's a Marine, and he's been like a dog on a bone with this problem. And he was the one who leaked the diffuse proposal. Are you familiar with the diffuse proposal?
No, what is that?
So there was a grant that was proposed to the DoD to go build a weaponized coronavirus under the banner of we need to build a vaccine against this because if we can do it, the Chinese can do it kind of thing. And the DoD turned down the grant because they thought it was too fucking crazy. You'll have something that'll leak from this. But as anyone who's written grants, I've been involved in writing grants for our company. We got like $30 million in grants from the NIH. You don't apply with a grant not having done 80% of it. It's just you're looking for the last 20%,
right?
Because you just can't. You need the grant process is so grueling and so competitive that in order for you to succeed, you have to have all this preliminary data that shows like we can get X done. We've demonstrated this piece. We've demonstrated this piece. Now we just need to do this last piece. And in that diffuse proposal, they had used particular restriction enzymes. These are enzymes that cut at particular sequences in DNA.
Okay.
Those are important because they help guide the way that you cut and paste DNA together. So imagine if you're cutting and pasting a Word document, but your highlighter to highlight what sentence you want to cut can only start and end with certain like letters. Those are what restrictions enzymes are. So imagine you could only cut and paste where there's a the. So you would have to then build a paragraph knowing that the paragraphs that have sentences that start with the, we're going to cut with this enzyme. And the ones that have sentences that start with we have another enzyme with things that start with there, which is a couple more letters onto the, right? Mm-hmm. We're going to use those enzymes to cut here. And by doing that, we can assemble this genome. Well, they had certain restriction enzymes in that diffuse proposal that are present in coronavirus that's currently circulating. And that was kind of the smoking gun that showed that, okay, this was in fact assembled. It was a product of the diffuse proposal. Someone eventually carried out that grant application to its final conclusion. And that's what's actually circulating. And
holy shit.
Rixey has the whole story on this. You want to talk to him and Alex Washburn. Alex Washburn did some really interesting work mapping out the frequencies of these cut sites in all other coronaviruses. And only in SARS-CoV-2 do they exist in a manner where they are almost perfectly spaced into. They build the thing back together in 6 pieces. So the Humpty Dumpty comes back together in a sort of an engineering way through only in SARS-CoV-2. If you look at where those restriction sites exist in all the other coronaviruses we know about, they're kind of randomly scattered. They don't look like someone designed them in. Mm-hmm. When you look in SARS-CoV-2, they're very perfectly spaced such that an engineer put it together. And there's a particular set of them near the furin cleavage site that looks like it's designed to take that furin cleavage site in and out and mutate it and swap it around so that you can optimize this to actually hit ACE2 receptors. And what's remarkable about the zoonosis thesis is that this thing came out pre-optimized. Like when we first found SARS coronavirus, the binding affinity to ACE2 was already optimized. That's not normal. Like if this were a zoonotic event, the leap would mean it was unoptimized and would take maybe 5 or 10 years to become more optimized in the population. That's another signature that someone had to have a piece inside the design of this virus that they could swap in and out to put a library in there to combinatorially screen for furin cleavage sites and receptor binding domains that would effectively allow them to enrich for optimization out of the gate. And so Charles and Alex have a good story around this about how they use serial passaging and the engineering design that they have in this implicate this as being, in fact, a weapon that was built. And it's not something that hopped over from pangolins and bats and all that stuff.
Jesus Christ.
Yeah. Harvey Rich, I think, has another interesting window on this. Like it's worth a view on the Ron Johnson thing that I was at. Harvey gave his pitch on like, all right, where the hell did this all come from? And how do we avoid doing this again?
Right.
And he had the most compelling synopsis, I think, because everyone's been like, can they be this evil? What the hell happened? And I think his story makes the most sense, which is that the biodefense, industry, if you will, has always kind of justified itself on that we need to be doing gain of function research to protect against a particular weapon. And so we're going to preemptively do all this. Here, fast forward to 2020, they've never come up with something they've saved us from. And then the first thing that leaks is potentially from their own making, right?
Right.
So that puts the biodefense industry on their heels in that we have to be in control of the solution to this. Otherwise, we're non-existent when this is done. So let's get Fauci involved because they had him on payroll. And of course, Fauci, I think you've probably seen some of those emails and conversations that have come out where it's like,
God.
You'll have orders, you have things to do.
He's so arrogantly talking about you'll get a vaccine or you'll lose your job.
Yes,
the kids will get kicked out of school.
His emails internally that leaked out of the NIH were like, you'll have things to do tomorrow. You must read this paper. This is a paper about, potential leak, right? Early on, this is like February and the pandemic where it first hit on him that this could have come from money from his pocket. Like it could have come from the WIV, the Wuhan Institute of Virology. And so suddenly Tony's now implicated in maybe have funding the virus that leaked. And suddenly he's in panic mode, sending emails to everyone that night, like, read all this stuff. You'll have assignments tomorrow. Like, we got to get on this. And Francis Collins is in there talking about how we have to have a swift takedown of all these conspiracy theories that say it could be engineered. And so they're in high gear right now trying to cover this up. But his take is that the biodefense program then went into high gear trying to get seen as the only solution to fix this. That way they would have a justification for, yes, something leaked, sorry, we don't really know who made it, but we happen to save the day with our biodefense program having this vaccine ready. See, this gain of function stuff really needs to happen, otherwise millions of people would have died without this. Which is a narrative you hear people try to propagate in the news, right? That without this vaccine, it saved 20 million people. They conjure up all of these weird epidemiological studies that try to make that case, and they're all modeling studies that are easily dismantled. But I think that is a lesson for us going forward, that this gain of function work is more dangerous than it can ever help, and it is something they need to put a real tight lid on, because this. I'm involved on a separate side of this, which is, okay, you found a contaminant in these vaccines, that could implicate fraud. Fraud might unwind the PrEP Act. Therefore, lawyers can now go after the FDA. And the first trial that tried to do that was with Brooke Jackson. And Brooke Jackson had evidence of the trials being fabricated. Like she was at Ventavia being asked to shift around the PCR numbers or shift around the data on unblind this site and blind that site. That way they could play games with the clinical trial data.
Right.
And she blew the whistle on that. They brought that to court. The judge ruled that this was not, yes, Pfizer lied, but they were told to lie by the DOD. This is not a typical manufacturing arrangement. This is actually a biological countermeasure. The DOD is running this. You have the wrong defendant. So they have to re-steer their case maybe toward the DOD. I mean, they don't know. This is, how do you sue the DOD?
The judge said that they were told to lie about the SV40 because of
not SV40. This is just fraud, a different form of fraud.
Different form of fraud. Okay.
But we expect if we approach them with the SV40 thing, they'll probably use the same excuse. Like it's a precedent that they've set.
I can't believe this SV40 story isn't everywhere.
Yeah, it's they have a lot of fact checkers talking about it, which is a sign that they're worried about it. Like they don't assign all the fact checkers you see. If you dig into them, there's some connection of money either back to the Gates Foundation or to a pharmaceutical company one way or another. Like they are pumping out. I mean, even the government came out saying they had $1 billion to spend on reducing vaccine hesitancy for basically marketing their narrative, if you will. So when you can tell which topics worry them by how many fact checkers are sick on it. And we're probably right up there at the top. If you look up the SV40 stuff, they have gotten every one of the fact checkers to write something about it.
What was the purpose of the Human Genome, or what was it for people who don't know?
Well, all right. So yeah, I'll back up on that. So we wanted to, the whole it was really, this is important history actually that you cover. I'm glad you asked. So the goal back then and the dream and how it was sold to the American public was really, ironically, through Francis Collins, who we'll probably get to later. And the pitch there was, we want to go to personalized medicine. We want to sequence everyone's genome, figure out their differences, and drug them accordingly. Instead of this, let's build one drug, test it on 10,000 people, and send it across the entire globe and hope it works.
Right.
The idea was, no, we're going to get more precision about this. We're going to actually, when you have cancer, sequence your genome and then sequence the genome of your tumor, which will be different in most cases, and see if we can find druggable differences that will kill the tumor, not kill the patient. And some of those started to occur in that time frame. There was one drug called Orissa that was specific to, it failed as a drug until they discovered, I think it was Dan Haber up at MGH that did this, and Matthew Myerson, Dana-Farber. They discovered that if you took that drug and actually looked at people's EGFR receptors, there was like a 15 base pair a deletion inside that gene that if you had it, the drug worked, and if you didn't, it did not work. So the drug failed on population-wide. But if you isolated the people and sequenced them first, you had remarkable success rates with the drug. So it rescued a drug just by knowing the genetics of the population to put it on.
That's amazing.
So that was really the whole goal of the Human Genome Project, was to do that 1,000 times over and do that in every disease, is can we target these drugs so that we're not sloppily blanketing the population with drugs that we test on small populations and they blow up on us when we hit different haplogroups or different populations out there.
And is it true that the variation in the human genome, there's not a lot of variation?
If you want to compare it to cannabis plants, we're completely inbred and should be walking backwards with stones up our nose. Yes. Yeah. So, maybe there's a million variants maybe between any given individual.
So it's like a variant for every 1,000 or something?
Yeah. Yeah. It's a fair number. And, now that we have better sequencers, some would argue maybe it's more like 1 in every 500 if you include some of these insertions and deletions and longer structural variations that we couldn't see very well back then with the technology we had that initially did the Human Genome Project. But 20 years on, as we are now, we now have completely different technology to survey genomes that are remarkable compared to what we had before, and we can see much more variation now than we could then. But still,
really?
Yeah, we're not at the level of cannabis plants. All right,
Right.
Cannabis plants are their own beast.
Now, is that just a quick side note on the genome stuff? Is that normal for most mammals?
The polymorphism rate that's a good question.
Because I was listening to something where you were talking about, like, essentially if you took our genome, if you extrapolate into the past, it basically means we came from like 10,000 humans.
Yeah, mostly in sub-Saharan Africa.
And there was like a bottleneck in the population, in the human population.
Yes, as Neanderthal came out, and the Denisovans played a role in some of this. So they're still piecing some of that structure together with paleogenomics. So there's a whole field where you can go back and sequence, things that are old. Like I was involved in one sequencing Utsi, the mummy, which they found frozen in ice that was probably 5,000 years old up in the Alps.
Oh, really?
Just to understand, what were the genetics like back then? I think they, interestingly enough, found Lyme disease in that guy. That's
really?
That's something that shocked everyone, because everyone believed that was a New World.
And this guy was from 3000 BC?
Something like that. Yeah, it's about 5,000 years ago. Yeah, that sounds about right. There's another Pablo. What's Pablo's last name? There's a really good paleogenomicist I'm forgetting, but he actually, I think he may have even got a Nobel Prize, so I should know this, but it'll come up. But he has done a lot of work in developing the methods that make it possible to sequence DNA that's that old. Because when DNA gets that old, it tends to break down in a particular manner, in a predictable manner, and you have to be aware of that in order to sequence it. And he's been very good at getting like woolly mammoth to sequence and a variety of old things, so you can begin to
this guy
put that back together. Svante Pabo, there he is. Yes, I forgot his first name. So yeah, he's a real pioneer in being able to do this type of work. But that helps bring the picture back. But you're right, we are a diaspora that came through a bottleneck, and which is one reason why many people on the Genome Project were. I think there's a strong message in here that this idea of race differences is actually irrelevant when you look at the genomes.
Right. Do we have any idea when that bottleneck occurred?
I think there is probably 100,000 years ago was when the diaspora was most extreme. Although you can find obviously hominids back a million years. There you even go as far back as Australopithecus. We haven't gotten sequence from material that's that old. I think some of the records in sequencing are probably 50 or 100,000 years. I haven't kept directly up to speed with it. Svante probably has the record on the oldest thing ever sequenced. I haven't kept up with the human population genomics story there because in some ways it's very boring. It's as you said, there's not a lot to it. We all came from a fairly recent ancestor,
right? And
it's you can find some interesting things about the genetics that change with lactose tolerance based on when we think we went through when we began to get involved in agriculture and a more farm-based lifestyle. So there are some genetics that follow that. But
there's also people like this that study climate change through like the end of the ice age around 11,800 years ago when there was this younger Dryas impact hypothesis where they impacted these comets came and hit like the North American ice sheet and wiped out all the megafauna here. But I mean that wouldn't be enough destruction, and that wouldn't be enough to bottleneck the population down to 10,000.
No, I mean the thing that's really unique about humans is that we're very adaptable and we can move. And as a result, I think you'll find that our genomes aren't nearly as complex as plants. Plants are sessile, so they're stuck. When the environment changes, they can't run away from it. They have to carry a lot more genomic tricks in their bag to compensate for it.
Right.
So in fact, there's a very good. An interesting piece of work I'd forward you to from David Sinclair, who works on a lot of this longevity stuff.
Yeah.
He was digging around in the xenohormesis field, which I think is fascinating. It's kind of an intersection of cannabis and human genetics, and that plants, as we became agriculturally centered as a society, and this has probably happened long before that occurred, but plants have this capacity of making secondary metabolites that signal to the organisms that eat them and spread their seed around. They give them information about the environment. So a lot of the secondary metabolites that you'll find in plants when they're stressed will signal to the organism that consumes them to prepare with caloric restriction. So you'll see many of the compounds that hit the rapamycin pathway and the mTOR pathway are polyphenols and flavonoids and things plants create when they're stressed out.
Whoa!
So this is one of these. This is something we pay attention to in the cannabis field, because if you want to get very diverse chemotypes of plants, you can't grow them in perfectly happy environments. Like you actually want to stress them to some extent so that you get anthocyanins to come up. When you stress them, you tend to get different terpene profiles. You tend to get the cannabinoids are coming off that same pathway.
And how would you stress them?
Well, in some ways, you never let them see pollen. That makes them stressful.
Okay.
So they end up growing really large flowers waiting for pollen. So the way to get the highest THC loads are to never let them see pollen, at least for the females. And you can also do it with light cycles. You can do it with temperature and pH and other types of treatments in the soil that will alter its actual secondary metabolite profile, so you get a different chemotype for the plant. That might be important in the way people are making oils for cancer. Like cancer is oftentimes a scenario where you need to grab onto the mTOR pathway or the rapamycin pathway and just change the metabolism of the organism, and you need to feed them with these extracts that are from plants that are stressed.